So welcome everyone to our sixth week. This will be our macroinvertebrate identification session. So those of you that joined us in week one, this is sort of our full circle around to actually being able to identify macroinvertebrates. I've already taken a look at some of the images and it looks like people are having fun out there, staking out their bags and sending in videos and everything else. So thank you for that. As a reminder, you are now part of the Center for Great Lakes Literacy. And we have our folks here, same as always. And I just wanted to acknowledge that everyone is still here and present, and thanks for a great teamwork pulling our 6-week workshop together and getting it going. And the workshops are being recorded. And there is, the chat content files will be available to participants for each week. And you will find your blank copy of the action form and all of the other information that you can see here. This is information that we've been sharing every week, so this really just serves as a reminder for you. We do have a little bit of new information today that I would like to share. So we do need a copy of your action plan. Some people have contributed them already, so thank you to those who have already submitted it. You did receive a link to the Google Drive folder, and in that folder you will find not only a copy of the blank action plan, but then you can also just put your action plan in that folder, so they're all contained in one place. CEU certificates, you should have received an email about this. The short of it is that if you are interested in having CEU certificates sent to you for the workshops that you participated in, then please email Cole. And Cole, if you could put your email in the chat, that would be helpful, I think. And let's see, what else do I have to cover today? The stipends, the criteria, again, we'll just quickly review. In order to be eligible for a stipend, you must have attended at least 4 of the 6 workshops, and you must have filled out your action plan. If we do end up with more action plans and folks who have met those criteria, then we will roll them out in order of registration. So if you registered early, then you have a greater likelihood of receiving a stipend. And just know that we're working on figuring out and identifying how many stipends we can actually provide. And we will notify you via email if the criteria have been met and ask for your preferred mailing address, and we'll mail you a check. And so that's how the stipends will work. If you have any questions, please feel free to contact me if you're a Minnesota educator, and Ann if you are a Wisconsin educator. We also have a post-event evaluation for you today. I will put that, since it's not technically the end of the workshop yet, I'm not going to share that link yet, but we will share it out right after the workshop and before the joyful hour. So you will receive that and also receive it in an email as well. Please do take the 20 or so minutes that it will take to fill out the post-event evaluation. This is probably the most important and way that you can be helpful to us for putting on more programming in the future. Is we rely on your feedback to be able to report back to our funders, and also we use it as guidance for creating workshops in the future. And I think that is about, two other tips that are specific to this workshop. This is a piece that we didn't communicate during our reminder email. If you have live macroinvertebrates that you want to show today, one good tip is to grab some ice chips during your 5-minute break and plunk it in the water that they're in, so it kind of slows them down because they can be a little bit zippy. And then if you have any macroinvertebrates to show and tell, if you could put your name in the chat, that would be helpful, because what we're trying to do is distribute evenly between the 3 groups the number of people who have macroinvertebrates that they would like to show during our small group work. And so with that, I think that's all I have. I would invite the other workshop organizers, if there's anything else that we need to say or anything that I missed, I'd invite you to mention it now.
So Marty, you were, just to clarify, you'd like them to put their name in the chat like ASAP, right? So that Anne can start sorting through who has stuff to share?
Yes.
Yeah.
If you know that you have some, or you're reasonably confident that you will have some to share, put your name in the chat now so Anne has time to work on breaking you up into small groups. Okay. And I think with that, that is what I had to share. And so I would just, I'll stop sharing my screen and I'll welcome Lee, and just excited to see what you guys have to show us today.
Are you turning it over to me? You broke up there.
Yes, I've turned
Okay.
it over to you. I stopped
All
my
right.
sharing.
Sorry about that.
Take it away, Val and Holly.
All right. So I'm going to give the presentation. Holly's getting set up over there to be able to augment what we do with stuff through the microscope. And I'm going to stop sharing my video as soon as I start sharing my screen, just because we do seem to be having bandwidth problems. And as I get started here, before I go into presentation mode, I want to show you that, come on, okay. The speaker notes on all our slides, which we are going to make available to you, have identification notes in them now. And so I think Holly wanted me to update one thing, and I'll re-upload the file after the meeting today, but you can download this. We'll have it available as a PowerPoint or as a PDF, so it's easier, and you'll be able to see all the identification notes. So I want to figure out how to stop video. And I apologize for the mask, but Holly and I are in the same room, and so we don't have a lot of choice about that. So hopefully you can hear me okay. I know it's going to be a little bit muffled. All right, so how do you identify macroinvertebrates? So we're going to start with the basics. I know that some of you are much more advanced than this, and I apologize for that, but I don't want to leave everybody else in the dust. So remember that many aquatic invertebrates are the immature stages. Some of them are just called immature. Some of them, like this one down here, are actually called larvae. And so they don't resemble the adults in many cases, and that gets really tricky. So what you want to look for is, does it have true jointed hard legs that could be used for walking, like this stonefly here, or this mayfly? Hard to see on this little caddisfly, but he even has some jointed legs. This wormy thing, which is actually a fly larvae, does not. It's got little pro-leggy thingies, but they're not jointed, articulated legs. And so you have to distinguish, does it have any legs at all? If so, what kind are they? Are they like little fleshy pro-legs that you couldn't really use to walk with? Or are they decent legs that they could use to walk? How many of them are there on the thing? Because remember, insects have 6 legs, but you can get things like mites and maybe even spiders that have 8 legs. And then this one will have 2 little pro-leg thingies. Can you see gills? And where are they located? And what do they look like? This can be really important. Stoneflies, you can't see it on this picture, but some stoneflies have gills underneath their armpits. So you have to kind of flip them over, look on the underside, and see. You see the gills here on this mayfly. Sometimes you don't see any gills. A lot of aquatic invertebrates get their oxygen just by it diffusing through their skin. Does it have a shell or a case? Obviously, this guy, this little caddisfly, has built himself a little case that looks like a snail. Do not destroy the cases that the critters come in. Oftentimes the cases are very key to helping you identify particularly the caddisflies. And then when you get into the stuff that's harder to describe and is more just, okay, it's long and skinny, It's really important to figure out if there is a hard exoskeleton or if it's just fleshy. And so I say, is it squishy or is it crunchy? If you just take your little tweezers or something and poke at it or crunch it a little bit, is it squishy, all fleshy, or is it crunchy? Does it have an exoskeleton? And that's important in a lot of the keys. So those are some real basic things that you're going to have to go over. Oh, I should also say, what comes off the hind end is really important. And so these are cerci, often called just tails sticking off the hind end. Sometimes things that look very similar coming off the hind end are actually gills. And so you'll need to use your magnifying glass to take a closer look. Sometimes you have to turn the insect from side to side to see that. And so taking a hard look at the hind end can be pretty important. Okay, so we're going to start with what we call the big three that are used a lot in trying to figure out if, especially a stream, is in good condition, but it works for wetlands too. So lots of people, especially fisher folk, have heard of mayflies. This is the famed Hexagenia, one of the, I think, the smaller variety. But the nymphal stage looks much different. And you see here that most mayfly nymphs have 3 tails, 3 cerci. Now this can get tricky because they get knocked off super easy. So a lot of times you pull your nymphal mayflies out and they have no tails. And how do you know they were supposed to have 3 tails, right? So that gets tough. And then there are a couple of groups that only actually have 2 tails, which would make them superficially resemble a stonefly, because stoneflies only have 2 cerci. So there's a little trickiness to it. So the thing is, if it has 3 tails, and it generally resembles this with 6 legs and maybe gills or maybe not gills, but if it's got 3 tails, you got a mayfly. So that's the easy part. The adults, of course, are winged. They live much less time, only a day or 2 than the larvae. But in your leaf packs, I'm thinking you would only have the nymphal stage. Stoneflies are another one of the big 3. And again, as I said, the larva have, or the nymphal stage has just 2 tails. The tails tend to look, the cerci tend to look a little more robust than on a mayfly. They tend to survive a little bit better in sampling, so they're less likely to get knocked off. Stoneflies are really active predators, the nymphs are. And so they have really robust legs. They're good at scrambling about. And there's one kind of stonefly that's not true for, and I think I have it on the next slide. But in general, they're really active predators. Many of them have their gills in their armpits, and you can just see those here. They look like a bunch of tubes, and if you flipped the stonefly over, you would see them better. This is a very common type of stonefly that we see a lot in streams. They're active predators. They wouldn't necessarily go into a leaf pack to eat the leaves, but they might go into the leaf pack to eat the other critters that are eating the leaves. In contrast, the adults are weak flyers. They mostly are crawling on the vegetation around the sides of the streams, and they can be quite large. But people don't notice them very much because they tend not to fly very much, because they don't fly very well. Shoot, and I didn't put a Pteronarcis stonefly in. So Holly's going to show you later a stonefly called the Pteronarcis stonefly. And these are very common in leaf packs. They're much darker, they're almost black, and a little more what we call roach-like looking than the common stonefly, but they shred leaves for a living. And up here in the north, it takes them 3 years to go through their life cycle to become an adult. So you'll find the littlest ones, and then the second years that are a little bigger, and then the third years, and the third years are very large. They can be 2 inches long. Holly's trying to get them up on the microscope now, but it doesn't look like we've got a great image. So maybe we'll show that a little later when she's got a second to work on that. And the final one of the big 3 are caddisflies, Trichoptera. Again, larvae look very different from adults. The larvae, almost all the larvae make some kind of a case. Very few are free living. They make cases out of stones or bits of vegetation or little pieces of sand. Some of them spin nets, so that's not truly free living either, although you wouldn't necessarily get the net in your sample. And then there's a predatory kind that is truly free living and is bright green and runs around. And but the key with caddisflies, so again, don't destroy the case. Really important. The cases, a lot of times you can key them out just with the case to family. And also, if you're going to try to remove them from their case, you'll probably rip them in half. It's really hard. They have little hooks on their hind end that hold on to the case so that they can't be pulled out of the case, because that's a anti-predator way of protecting themselves. So what you do is you take the end of the case, the back end of the case, and you squish it gently several times in several different directions with your forceps, and then you may be able to use 2 tweezers, one to hold on to the case and one to hold on to the critter, and you may gently be able to pull it out. Much of the identification of Trichoptera relies on the case itself. The adults look like little moths that live along the stream edges. So if you see things that look like moths flying around stream or wetland edges in the daytime, those are actually the adult caddisflies. And the way you tell the difference is that moths, when they rest, they hold their wings like butterflies, right? But Trichoptera don't, caddisflies don't. They hold their wings like a tent, an A-frame tent over their backs. So that's the way you tell the difference. We think that you will find Dobsonflies and Alderflies in your leaf packs. Again, these are really common insects in leaf packs. They can be quite large. The larvae are impressive. They can be an inch or several inches long. Here you have to think really hard. So your legs are the segmented legs up here on the thorax, and then these tubular things are actually gills on the abdomen. So again, 6 legs, it's an insect. These are gills down here, and then notice we've got some prolegs coming off the hind end here on this one. Whoops. Wasn't done yet. This species has more of a long tail-like thing, but again, you see the tubules. So people don't know much about Dobson and Alderflies because the adults are weak flyers that crawl around the vegetation. So a lot of people have never heard of them. But they are reasonably common in good condition streams and streams with lots of leaves in them. So you may find them in your leaf packs, or the kids may. You're going to find all kinds of fly larvae. It's pretty amazing how many kinds of fly larvae there are. So we're going to spend a few minutes on flies. It's going to be flies, more flies, and more flies after that. Those of you that live in the north may be familiar with noceums. They're called biting midges or ceratopagonids. And believe it or not, the larvae, this worm-like thing, are also predatory. I mean, who would believe this thing could be a predator? But it is. Not very fast-moving predator. It's got little tiny prolegs. It's got a hard, what we call sclerotized head. So the head capsule is hardened. But the body itself is real squishy. And you see it's very long and rope-like. So you're going to see a bunch of these fly larvae that are just generally long, tube-shaped, white, squishy things. They can be quite hard to tell apart. We'll give you a few tips and tricks. It can get hard even for us sometimes, and you need good magnification. So you may get stuck at, okay, it's got some prolegs, it's got a hardened head, it's probably a fly larvae. Another one that you're going to find almost certainly are Chironomidae, the non-biting midges. So this is an adult. If you look at it, it looks like a mosquito, but it doesn't have the long proboscis that a mosquito uses to bite you. So that's why it's called a non-biting midge. It won't bite you. The males have these bottle brush antennae. People see them all the time and think they're mosquitoes. There'll be giant swarms of them, mating swarms along streams and lakes and rivers and wetlands. The larvae may or may not be red like this. This is hemoglobin. It denatures very quickly when put into any preservative or when they die. So you pretty much only see them in this red color when they're alive. But this C shape that they're in, laying on their sides, is really characteristic. Now these guys are little. They're going to be, you know, a 1/4 inch long and a little thicker than a human hair kind of thing, or a 1/2 inch long and a little thicker than a human hair. So they're quite small, but you're going to see a lot of them. Again, they have the hardened head that you can just see here. They have little prolegs that we call parapods. They also have little prolegs on their hind end that's a little harder to see. But the C shape is characteristic, and you could get dozens, if not hundreds, in your sample. There's a billion different species of them. And I'm probably not exaggerating too much. They live in all types of habitats. If you don't find chironomids in any kind of aquatic invertebrate sample, it would be really weird. And I would just say you're losing too large a mesh size, because they're there, they're just going through your mesh. Stream samples may or may not come across black flies. I'm not going to spend any time on them. They're pretty characteristic and easy to see, and you wouldn't necessarily get them in a leaf pack. What you will get in a leaf pack, you should get, are cranefly larvae. Cranefly larvae look a lot like your typical grub, except they tend to be a little darker in color. And again, we tend to call fly larvae grubs. So these are aquatic fly larvae, they're grubs. The trick with the Tipulids, the cranefly larvae, is their head is kind of sucked up inside all this fleshy stuff at their front. So you can't really even see their head. You don't know where their head is. This interesting stuff on their butts is gills and things, and is used a lot in taking them down to genus. But they're big, fat, grub-like things. And so if you find these big fat grub-like things in leaf packs, you've probably found a cranefly larvae. We're familiar with the adults. They look like humongous mosquitoes. Some people call them mosquito hawks. They don't eat mosquitoes, which is more the pity. But they're very beautiful, very patterned wings. They're really attracted to lights. They don't bite. Don't worry about it. As I said, flies, flies, and more flies. Water snipe flies are something that are very common in northern streams. I get them a lot in leaf pack samples and just in leaves. And the trick with these, okay, so they've got a real characteristic shape. They come to this point, and they're kind of, you can kind of see their nice segments down here. And then they have these two tail-like things that stick out in a V shape and are covered in hairs. It's really characteristic. They're really easy to spot. So you see something that looks like this, you've got a water snipe fly. They tend to be fairly large, 1.5 inches long kind of thing. Horse and deer flies are always interesting. They're not nearly as common in samples as water snipe flies. Again, grubby looking larvae. The larvae are really hard to tell apart, but I just wanted to show you because everybody knows about horse and deer flies and how nasty they are and how hard they bite. And it is possible that you will find the larvae in your samples. You might get mosquitoes, especially if your students put their leaf packs in ponds or really small bodies of water that don't have fish. So again, they're flies. These look a little bit different. They have the bigger, brushier antennae. Sorry, these are mouth parts, labial mouth parts here that they use to feed. Some of them have this characteristic tube off their hind end with the hydrofuge hairs that they use to go to the surface and breathe. Others don't have this tube. They just have the hydrofuge hairs right here on their rump. But the brushy antennae and the general shape are helpfully characteristic. They also wiggle in a particular way, and mosquito larvae are called wigglers because of the way they wiggle. I think you're all familiar with the adults who love to suck your blood, and they do have a proboscis. They don't have the bushy antennae like the non-biting midges. Dragonflies and damselflies. Don't know if you'll get any in leaf packs, but they're so much fun, we just had to talk about them. So the dragonfly and damselfly larvae are pretty voracious predators. This is a dragonfly larvae. These are his mouth parts. These are these teeth, these snapping jaws. The jaw is hinged, and they can sort of shoot the jaw out and grab things like little mayflies. It's really impressive. And if the kids can get a view of some of these these with their teeth, they'll be suitably impressed. Magnification, of course, is needed, but even at 5x on some of these bigger specimens, look at the size of this eschinid here, you can see them. So the more mature larvae will have little wing pads coming off their back, but the shape is very characteristic. And I'm going to go into how to tell dragonflies from damselflies in just a second, but you see big legs, big eyes to hunt with, this hinged jaw. You have to flip them over to see how it's hinged and use your tweezers. Dragonflies have gills up inside, so you can't see any gills. And here's one that's caught a water strider. So they're voracious predators, as I said. So how do you tell a dragonfly from a damselfly? First, the adults. So dragonflies are just generally more robust, especially the hind wing is pretty large. It comes down. And when they rest, they hold their wings straight out. Damselflies are much weaker flyers. They're much thinner, they just look more delicate, and when they sit, they hold their wings together over the back. They do not hold their wings out. For the larvae, which is what you'll likely find in your things, one of the easiest things to do is to look for gills. Now again, the gills are really easily knocked off on damselfly larvae, but there should be gills coming off the hind end. And there should be 3 of them. They tend to look like leaves. So you can tell them from cerci, from tail, so it's not a damselfly. Sorry, it's not a mayfly because it's actual gills, not thin rods that are just cerci. And so again, they may be flattened, but if you look at them from side to side, you should see that they're broad in one dimension so that they can collect oxygen. Dragonflies don't have any gills that are visible. They're up inside, and dragonflies tend to be thicker and more robust. There are other clues, but the easiest thing is just to look for the gills. Now the problem is, what happens how do you know that you've got a damselfly that didn't just get its gills knocked off? And that is more difficult. Again, the shape and the robustness, and there are a few other things in the keys that can help you. We don't know if you'll find true bugs in your leaf packs, but we think you might. Did want to give you a warning about this guy, back swimmers, Notonectidae. They have a proboscis. You can't see it here because it's tucked under their head, but they have a bite that's like a sting. The way they feed is they stick their proboscis into their prey and inject digestive juices and start to digest their prey from the inside out, and then they suck out the digested stuff, and that's their food. When they do that to you, it hurts really, really bad. And I think they must also inject something that must paralyze their prey. I did not listen to my professor, and I touched one with my bare hands, and it bit me, and it hurt like a wasp sting. So you don't want to touch these guys. They tend to be easy to spot because many of them are white, and white's not a super common color among aquatic invertebrates for something that's this big and moves like this. They tend to be white with red eyes, and I mean, they can be really red eyes. This one's preserved and it's a little lost its color, but they can be like glowing red eyes like a red Sharpie marker. So warn your kids, don't touch these with your bare hands. The other way to tell them is when they swim, they do literally swim on their backs with their belly up, like the name says. They lay on their backs and do the backstroke. They're really good swimmers. They're really good predators. Don't confuse them with these poor little water boatmen, which are vegetarians, don't bite anybody, and are really cute. You can tell them apart because water boatmen tend to have this really patterned black and white patterning on their carapace on their wings. So they may have red eyes, but they're not all white. They don't swim upside down. They swim right side up. And this real black and white patterning is characteristic of most water boatmen, and you don't need to fear these guys. Again, both of these are super good swimmers. They're not attracted to leaves, but you might end up with them in your samples. Other true bugs that you probably will encounter are water striders. There's 3 different kinds of water striders. I'm only showing 2 here. You're probably all familiar with these, the Jareds. They're the big guys, those great big water striders that skate across the surface. They're, a couple inches in length. The next size down are called the mesoveliids, the broad-shouldered water striders. And they're maybe ½ inch to 1 inch long, much darker in color. Many of them don't have wings. Oh, and so that's a thing with bugs I wanted to say real quickly. Let me go back to my other picture. The way you tell true bugs from beetles, and you'll need this for the key, is in true bugs, the wings crisscross like an X over the back, and the wings are leathery. And the immatures just have wing pads that aren't fully developed. And so in beetles, the wings are going to meet in a straight line all the way down the back, and they're going to be hard shells. And the immatures look like kind of like grubs, almost like fly larvae. Now, it gets trickier when this guy doesn't have any wings. Some water striders are wingless, but you can clearly see the segments here, and it's a water strider, so it's a true bug. All water striders are true bugs. So here are the beetles. We think you might encounter riffle beetles. Riffle beetles are really common in slow-moving water, and they're detritivores, so they're shredding up detritus and things, so they may be in your leaf packs. The adult is a very slow-moving, crawling thing, and here I want to show you, so these hard wing covers are called elytra, and it's harder to see on this one because these elytra are so patterned, but they meet in a straight line all the way down the back, and his wings that he flies with are membranous and tucked in underneath, and you would actually kind of have to pull the elytra off to even see them. But if you think of, if you've ever seen a ladybird beetle fly, they hold their elytra out like I don't know, kind of funny looking, and then they fly with their other wings. Their elytra are held out to the side and they look really weird. That's all beetles are that way. As I said, the larvae can be mistaken for fly larvae. The trick here is, remember I said you got to figure out if they're squishy or crunchy. So this larva has a hard exoskeleton all the way down and has 3 pairs, 6 real legs, not pro legs, actual jointed legs that it can use to crawl around with, and it's covered in an exoskeleton all the way down. And so that's critically important for the key. And so just don't be afraid to touch your critters and squish them gently, and poke them and see what they're made out of. Now, on the much more rapidly moving side of beetles, you have things like the predaceous diving beetle, which are really cool. I don't think you'll get any just in leaf packs, but they're way cool, so we had to talk about them. Again, hard elytra that meet in a straight line down the back on the adult. These guys are really good swimmers. You can see they've got swimming hairs on their legs, active predators. The larvae are impressive enough that they're called water dragons sometimes. They have, it's a little hard to see in this picture, but they have pretty impressive mouth parts. Not quite as impressive as dragonfly larvae, but pretty darn impressive. So have your kids take a look if you should get any of these. We don't catch them very often. They're not commonly found in samples, but they're way fun. Again, notice the legs. So you got 3 pairs of legs, marking them as an insect. Other things we think you might encounter. Snails, you can recognize snails. That's not going to be a problem. There are some kind of snails that look like little limpets. If you're familiar with the little limpets from saltwater, they're little cone shaped. They're very tiny, but those are snails as well. So if you get some little things that are like cone shaped and look like half their shell's missing, that's a snail. It's a limpet. We expect you to get lots of aquatic segmented worms, the oligochaetes, super common. It'd be weird if you didn't get them. So they're not going to be red. Again, the hemoglobin, not all of them make hemoglobin, and it denatures quickly. They tend to be white. They look a little bit like the non-biting midges, those white ones, but they're much longer, much more coiled. They look like a worm. There'll be these little white coiled up worms that are all over your sample, unless your mesh size is too big and you accidentally wash them all away, because they are very tiny. If you do get freshwater mussels, we ask that you and the kids try to put them back as quickly as possible. Many of our freshwater mussels are threatened and endangered species now, and so we try not to take these. Obviously, they're not zebra mussels, black and white stripes, triangular shaped. Most of our unionid mussels are quite large. They're the smallest that I've ever seen was the size of a silver dollar. So they, lay in the palm of your hand kind of thing. They're not going to be in your leaf packs. If the kids are out collecting and find other things, they might come back with these. Crayfish probably won't be in your leaf packs, but again, if they're collecting other types of samples, you'll find them. Be on the lookout for rusty crayfish. They have this rusty orange spot. It looks really dark here, but can be reasonably orange color in real life. We ask actually that you kill them. They're pretty destructive to the ecosystems that they've invaded. If you use a fine mesh size or end up with a lot of stuff in your bag, you may end up with mites in there. So aquatic mites are related to spiders. They have 8 legs. Hard to see, you have to really count. But they, and they're little tiny. They look like, I don't know, an enlarged period punctuation mark with legs swimming around. They're voracious little predators on eggs of other insects and critters and things like that. They're just lots of fun. This is obviously highly magnified. But your kids may point to little tiny things that are scrambling around with 8 legs and they really scramble. That would be mites. Some of them have blue gel-like translucent legs. They're super cool if you can get them under good magnification. We expect that you will find amphipods and isopods. They're very common, what we call detritivores. They like to shred up leaves and things to eat. So the way you tell them apart, so they're usually 1/2 inch to 1 inch long. The way you tell them apart is side swimmers, amphipods, lay on their sides with all their legs pointing in one direction. They have multiple swimming legs, 2 pairs of antennae, and they can be green or orange in life, tend to fade to white in death. But again, lay on their sides, have way too many legs to be an insect. And this is just, they all tend to look just like this. They're really characteristic. Isopods are related to sow bugs, and they don't lay on their sides. They have their legs sticking out in both directions, just like you would expect. And again, way too many legs to be an insect. They got bunches of legs that they use for crawling. They can be green, again, often fade to white in death. We have some that are gray colored as well, or tan colored. So again, just the shape and the orientation is one of the biggest ways to tell the difference. You might get leeches. Leeches can be very tiny. You get leeches that are way smaller than your little fingernail. Again, they'll have a way to tell them apart from other worms is they'll have a sucker disc on at least one end. Some have a sucker disc on both ends. You've got to see that on the underside. Turn them over so they're laying on their backs. You might need magnification. Unfortunately, if you put your samples in alcohol, leeches will shrivel up to really tiny round balls, some of them that can be just really hard to work with. If kids are just collecting water samples, they may get the really super tiny stuff, our zooplankton. So you get the water fleas, which are the Cladocera, our copepods, and our seed shrimp, our ostracods. These are just barely visible with the naked eye. The kids can see them better than us because their eyes are so much better. But I put some identification notes in there, but I really don't think this is something you're going to be dealing with. But should you have access to a microscope and get water fleas, they are super cool. If you put them on under a microscope slide with plenty of water under there, they'll lay on their side and you can actually see their heartbeat if they're still alive. And you can see the babies in the brood chamber. This is an egg, but many of them will have live babies in their brood chamber. And you can see the little tiny ones. So they're super cool if you have access to that. It's a fun thing to show the kids. And I think, yep, that's all. So I'm gonna try to stop sharing. And Holly's getting set up over there. Did you get a Pteronarcis? Okay, so Holly's going to show you that. You want to share your screen, Holly. She's going to show you that Stonefly. And Holly, you'll have to unmute too, I think.
Hey, Val, one quick question.
Sure.
Are rusty crayfish edible? Could we eat them?
You can, and they could get pretty large. We, in other years, have, non-COVID years, have done crayfish boils with rusty crayfish. There are I don't know, millions of them in the white iron chain of lakes up here and into the edge of the boundary waters, and people put out crayfish traps and catch thousands of them and do a crayfish boil.
Perfect. That's a great way to get rid of invasives.
Oh, absolutely. Okay, so Holly is showing Pteronarcis stonefly. Unfortunately, they're so big, we can't hardly zoom out enough under the microscope. But they tend to be darker in color than other stoneflies and a little more roach-like in look. And as I said, they can be very large, and you can see the little gills here. But want to give time, and this one, of course, it's lost one antennae. It's supposed to have one antennae here and one antennae there. So that's what happens. You know, you get stuff, and just in the handling process, it's gonna lose legs and antennae and gills and so forth. Oh, she's flipped him over, and so now you can see all the tubular gills in the armpits. Yeah. Oh, right. So she's pointing out that can you focus that a little more? If you look at the end of the leg, one of the legs is sticking kind of up towards us, and it has two claws. And she's going to use her pointer again to show you here in just a sec. It has two claws on the end of its leg, and that's different than mayflies. Mayflies only have one leg. Holly is on a dissecting scope with did we figure that was 10x? 1 point. Yeah, it's about 10x magnification. So Pteronarcis stoneflies are so big, you can easily see most of these features with a magnifying glass, or the kids can probably see them with the naked eye. So if you are lucky enough to get Pteronarcis stoneflies, they'll be good. So you can do a ton of identification with a hand lens and 5 to 10x magnification. You easily get to order, probably to family. Often it's more the terminology that's the issue than it is the magnification. Yes, there are invertebrates that take more than one season. In fact, up here in the north, it depends on water temperature. So the farther south you go, the quicker they can get through their life cycles. The farther north you go, the slower it is for them. So these pteronarcids up here, they take 3 years to go through their life cycle. Farther south, like in southern Michigan, they probably only take 2 years. Farther south than that, maybe they get through in a year, I'm not sure. Hexagenia mayflies, up here they take 2 to 3 years depending on the water temperature of the water body. But then there's things like midge larvae in warm wetlands. Midges in warm wetlands, the flies, they probably have multiple generations a year. So it all depends on the critter and the location. And somebody can help read the chat because I'm probably not going to get through.
No, you're doing a great job. There have been a few questions to this point, so you've got them. So.
Okay. Would the Chironomidae be wiggly? The Chironomid, when they're alive, they wiggle, they kind of flip around. It's I don't know how would you describe Chironomid? Yeah, it's kind of a wormy. Yeah. The other thing is a lot of the fly larvae, I mean, their only way to move is to kind of wiggle almost like a snake. They undulate. And so, so that's, yeah, so that's very true. Okay. We put some keys up online and some links to some online keys. So if you go into the resources that they listed, the link for resources that was listed in the email, you will find keys that we put up there, and also some online keys that have better pictures. Holly and I also are willing for you guys to email us and send us pictures, but I know that it's really hard to get decent enough pictures of these kinds of things. Let's see, why aren't deer flies and horseflies common in leaf packs? We just don't run across those larvae as much, and I'm not familiar. For deer flies and horseflies? Okay. Holly's talking to me, sorry. Yeah, so, yeah, we just don't, they might be in leaf packs, they're just not very common, which is surprising for, I think they may more live in ditches and smaller bodies of water that don't have as many predators. I know that's where I'm finding a lot of the adults are near my roadside ditch rather than the pond, the wetlands and other places where I work. So it may, yeah, so we don't find them nearly as much in streams. So if your kids are putting their leaf packs in wetlands, you may find them more. Okay, by what you find, and what about the health of the water by what you find in leaf packs? So again, the number of different types of critters that you find is important. So areas with poorer condition will just have fewer things. I like to do a really crawly versus wormy type of indicator. Just if your sample is almost all wormy type things, even the flies that look wormy, and there's very few active movers in your sample, usually that's even in a leaf pack, usually that's an indication that there's something wrong. And things like these giant Pteronarcid mayflies that Holly was showing, they take 3 years to go through their life cycle. So having something in your leaf pack that has to take 3 years to go through its life cycle, that shows that that habitat's reasonably clean, reasonably stable for them. Otherwise, they wouldn't make it all 3 years and they would die. So those longer lived, more bigger things are another good indication of how the area is doing. And if you download my presentation again, you'll see that I talked about that more in the first presentation, and there's notes to that as well. What else can I answer for folks? Or we can start taking other types of questions. What I'd really like to answer too is if folks have been trying to use, sorry, this is Val, Valerie Brady. So if folks are trying to use the keys and you're getting stuck on terminology, or you don't know where to look to find something that's telling you to look for, or whatever, there are probably other people that are stuck at that same place. So if you have those kinds of questions, we'd be really interested in trying to take those as well. Oh, yeah, the preferred process for removing a leaf pack from a stream. So you've got critters in your leaf pack, you're hoping, and your leaves have been breaking down. And so the removal process, if you can, you want to be ready to, as quickly as possible, with as little agitation as possible, get that leaf pack into something from which the critters cannot escape. And so whether it's a bucket or a dishpan or anything, so you want to get, if it's safe to for the kids, you want to get as close to the leaf pack as you can, and then reach down, pick it up, and put it right in the bucket or the whatever else, so the critters can't escape. Now, obviously, if they've done the thing where, it wasn't safe to go in the water, and so they had to tie a rope to it and toss it out, you're going to have to pull it back in. There's going to be some critter loss, but again, as quickly as possible, put it out. Put it in secondary containment. I don't think mussels will end up in leaf packs. We just wanted to show the mussels. They're not going to get in through the mesh. So, but if the kids are in the streams looking at stuff, they may find mussels, and we just wanted to sort of reinforce that that a lot of our mussels are threatened or endangered species, and in many states, in many areas, you're actually not allowed to take them without a permit. And so just have the kids put them back. And, you know, they're going to be good size. They're going to be inches long. They're big buggers.
All right. Well, thanks so much for showing us some of the things under the microscope and for answering questions. It looks to me like all the questions that came in have been answered either by Val, or there's been some responses that Holly has typed in while Val has been presenting. And I think we still have a few minutes left of this portion. But Val, if you are in a space where it would be okay to transition into breakout groups, what I would recommend is starting that process just a couple minutes early, so folks have a little bit more time and to do small group work. Is that acceptable to you and to Holly?
Yep, it'll take me a minute to move across the hall so that Holly and I can both talk at the same time. And there's one more question here. The leaf packs will break down differently in different types of water, especially if there's just fewer critters. It will take them longer to break down. Remember, we talked about different types of leaves will take longer to break down. And if you put the leaves in an area where leaves are not common, some kind of a habitat that doesn't have trees around it, it may be that that takes a lot longer because there aren't the right types of critters there to break them down. More acidic water, like bogs, would take longer. Colder water would take longer. So yeah, you could get a lot of differences. And again, that's an interesting question that the kids could explore, different types of water. So I'm going to move across the hall and Holly, then give Holly this space.
Okay.
Oh, hang on one second. Oh, Holly wants to know, yeah, go ahead, Holly. So Holly's going to swap out real quick just before we do that, since we did take all this time to set us up. She's going to show you under the microscope some different, you can just leave that there because I think it only bothers you, some different types of mayflies. The picture didn't flip, Holly. That's weird. Oh, there it is, not went. Okay, so this is a heptagenaid mayfly. It's super flattened, can live in down in their stream current, and you can see its little gills here, but its head is really flat, and so that's a fun one. I'm going to move to the next one, Holly. And then this is what we call one of the minnow mayflies. They're baited. I think these are baiteds, right? It's a baited. See the three tails? Lost most of its gills, but they swim so well that if you saw them swimming in a stream, you would think they were tiny little fish.
That is really cool. So are there other mayflies to show us, or is that the...
She's going to try to show a couple of caddisflies really quick just because there's that diversity. It's okay. There's a little lag in the oh.
Val, did you notice there's one more question there?
I did. And so while Holly's switching to trichops, so dissolved oxygen is one of the main things that is driving a lot of the indicators that are used for streams in particular. And so in the old days, back when this was all figured out, people were dumping raw sewage and things into streams, and the bacteria eating the raw sewage would consume the dissolved oxygen really quickly, make the streams go hypoxic or anoxic, drive most of the fish away, kill much of the aquatic life, and only things with like hemoglobin could still live there. And so the EPT, the mayflies, caddisflies, and stoneflies are quite sensitive to dissolved oxygen levels, and that's why they're considered one of the major indicators of particularly nutrient pollution. So when you have excess nutrients, you have excess algae. As the algae dies, it's eaten by bacteria, and the bacteria use up all the oxygen in the water, and then you drive out your stoneflies, caddisflies, and mayflies and the fish because the oxygen's too low. So it all started with dissolved oxygen levels. Now we've moved into other kinds of toxic chemicals and toxic pollutants, and that's trickier because some mayflies are susceptible and some are not to these various things, and some stoneflies are susceptible and some are not. So it's not as easy to use the indicators anymore. So Holly's trying to show you some caddisflies that have been pulled out of their cases. And one thing you'll notice is that the sclerotization, the hard part, is just around the head and on the legs. And then the rest of the critter is mushy and soft. And so that's, and they're going to ask you about that in the keys. And so that's where paying attention. You have to use your pointer, Holly. I can't use it. So, and so that's where it's important to note the little spots that are sclerotized. They're going to ask you about that. And Holly's been pointing to the gills, the little tubular gills down the sides. So we probably have to go into breakout groups, but Holly's going to stay here at the microscope. And oh, there is one of the cases of the caddisflies. And it looks like a mess, but it's actually a caddisfly case that it built very carefully to protect itself. And those cases also, it can pump water through its case to help flow oxygen over its gills. And so here's some other cases. Again, always tell the kids, be sure to save the cases. Very important for identification.
And they're pretty too.
They are very pretty. I tried to make a necklace out of one.
Well, yeah, and there's jewelers that use that. So
exactly.
So thanks so much for all the time you guys put into setting this up. I think that we've really shown some amazing pieces here. I see there's a couple of other questions rolling in, but I think in the interest of time, we will save that chat, and then Val will ask you and Holly to respond via email so those other questions get answered. And
sure.
So let's take just a couple of minutes for a break so Val can get moved over into another room and so people can grab their macros and their ice chips and whatever else they might need. We'll be back in 5 minutes. This is a pretty large group. Our breakout groups are pretty large this week. We've got about 10. We're not going to do a bit.ly link and fill out a form this week. We can just focus on practicing identification. So do the things you need to do. Grab your leaf packet,
so you're going to have to
a bucket or
yourself.
a tub, and magnifying glass and your ice chips. And we'll see you back here in 5 minutes.
And those who are lucky enough to get put in the group with Holly, she'll have the microscope.
Okay, so Val, if we can get a screen share stopped now, we can flip up a break slide and be back. All right, well, with that then, Anne, I'll turn it over to you to get folks sent into breakout rooms, and we'll work on doing some ID. I'm pretty excited. I'm seeing some videos of cool, or cameras of cool stuff out there. So.
All right, ready to go. I'm going to send you guys now. And Marty said there's about 9 or so in each of the groups. So they're a little bit larger than usual. So have a great discussion.
There, that will be better. So thanks for being willing participants in that. We weren't really sure how it would go trying to do the virtual identification. So hopefully there was some value in there for everyone, whether they were learning that there were things that maybe should have been considered or communicated more clearly in the beginning, or whether it was things that, technical challenges or what have you. So hopefully someone got a little bit of something out of all of that. So with that, I don't think well, I guess I'll just ask the group, we've got just a couple of minutes here. Is there any, I'll just open it up if there's anyone who would like to share something from their group. We'll maybe spend a minute with this if there was an observation or a question for the group or something like that before we move into the last few details before we have our second break and then move into joyful hour stuff.
So Marty, I would like to share something that was really important that Val shared with our group. One of our participants had a bag near a pasture that that cows were grazing in. And so if you choose to use a stream near livestock, there obviously is an enhanced risk of E. coli in that stream. And so it's very important to be aware of that, to properly disinfect the equipment or anything else that was in contact with that stream. And obviously the kids should be washing their hands or perhaps even wearing gloves. So obviously you're going to catch different types of critters there in a high nutrient stream, but there are needed extra precautions if you choose to use a location like that.
A good bit of safety information. Thanks, Ginny. Anything else? Okay, hearing nothing, I think we'll move. Oh, best session. We had so much fun. Oh, good. I'm glad that it was fun. I thought it was fun as well. So a couple of things that we need to do to sort of wrap up before we transition into a joyful hour. The first, and this is probably one of the most exciting things, is we did say that if you participated in the macroinvertebrate sessions, that we would indeed have a drawing for a full-on leaf pack that we can then ship to you. And so Cole, if you're ready for that, I can turn it over to you in just 1 second. The other thing that I'm going to do now while Cole is getting that piece up and ready is I'm going to add a link now to our post-event evaluation, because we are officially done with our 6-week workshop series. We do have our joyful hour wrap up as well, but that's a piece that is but I'm going to share the post assessment now, so you guys have quick access to that. And so I'll do that. And then Cole, I'm going to turn it over to you for our leaf pack drawing. I don't know if we need a drum roll or something like that, but
well, let me share my screen here. So what we have done is, if you attended session 1 and today's session, we put your name into the raffle wheel of fortune. And so we are going to spin our wheel. Everyone can see that, right?
yes, I can.
Okay. So here we go. Drum roll. And spin. Yay! So it looks like Linda is our leaf pack kit winner. Thank you for joining us. We will work on getting that out to you by mail. I assume that we'll send it to your home address. Otherwise, let us know, and we will get that taken care of.
Okay, thank you, Cole. And I did drop the link to the post-event evaluation in the chat, so you have that now. And so for those of you who aren't joining us for the Joyful Hour, thank you so much for being a part of this 6-week workshop series. We really hope that you had as much fun with it as we did. And we look forward to connecting with you again in the future, a couple of times over the course of the year. So with that, I think I will say goodbye, everyone, and put up our break slide, and we will be back for our Joyful Hour beginning at 10:31.
And thank you to Holly and Val for 2 wonderful sessions with the macroinvertebrates. We really appreciate your time getting everything set up and getting everything ready. We know that it perhaps wasn't as easy as it might otherwise have been, having to share space and masks and everything else, and just really want to extend our gratitude for all of the work that you put into this to help us learn more about macroinvertebrates.
You're welcome, and I put my email address in the chat. Holly and I are willing to entertain questions and look at pictures and try to give pointers. We're still doing field work, so we might be a little slow getting back to folks, but with patience, we can do this.
So we have a little bit of time for a wrap-up session here, and so I'd be curious to hear from each of the groups on the things you talked about and some of the suggestions that were made. We had, how many groups did we have, Anne or Brenna? We had 3 groups.
Yep.
Okay. So let's see, I took notes for the group that I was in. Who took notes in the other 2 rooms?
So Marty, I also submitted the notes from before before I got switched. So yeah, I took notes for that group for the first half, and then for Anne and Cole's group after that.
Okay. Yeah, all of a sudden you were just sucked away. If that's okay, I jumped in and took notes, so it worked out fine. Okay, so do we have people who would like to volunteer to share out for the other two groups? I can share out for our group, but of the other two, do we have a volunteer who would like to talk about the things?
I could share out for the, I think it was Leanne, and I don't know what group is what. So, and Kate and Jennifer and Celine and Olivia was on and then disappeared. So, and I think it was a really good conversation. Lots of interest in doing the leaf packs this fall. I like the idea. Let's see, who was it? Oh, let's see, Celine, right, is going to, they've already got 100 leaf packs put together in Ziplocs or something like that, and are putting them, sending them out with the, with Chromebooks to the teachers or to the parents. And so that's their first kickoff activity for the year. And then there was a lot of discussion about iNaturalist and how it can be used with keeping some confidentiality in place by sort of widening your location that you're representing, or moving your pin slightly so that it would be more comfortable to have parents using iNaturalist without feeling like they're identifying their specific location, which I thought was an interesting thing everybody might want to think about. Also discussions about having a buddy classroom somewhere else so that you could compare some of the results you get with leaf packs and things like that. So you should all remember that idea. And we talked about interest in doing microplastics, and the fact that one of the Sea Grant people who are working on the Great Lakes could always be the collector of microplastic samples for teachers who are inland. And remember that you've got a really big resource in terms of microplastics and other things if you need to get samples from a different location, as long as we can get out and get them mailed to you or whatever. There was a whole bunch of other stuff, a lot of resources that came up, like the Madison Water Resources Library, UW-Madison, Great Lakes in My World, Coco Ross is a really good resource. What else, you guys? I don't want to take too much time.
We were talking about Liz Sutton from UW School of Fresh Waters, and she has some grad students that, work with different things. We didn't talk about this, but I was thinking maybe they could join, I'm no expert in any of this, but maybe they could join me in a Google Meet with some of our students and add a little bit of their expertise. So maybe inviting some of those students over would be great.
Very cool. Anything else from your group, Cindy?
Yeah, Leanne is waving her hand. Does that mean you'd like to share something, Leanne?
No, I was saying goodbye to Brian.
Oh, bye, Brian. He's gone. All right, so that's probably it from our group, and we can move on.
Okay, and so our group can go last. So whoever the spokesperson is for the other group, if you could share out, that'd be great.
If you want.
Sure.
So I was in with Cole and Anne and Molly and Olivia that's where she went. She came to us, I guess. Because Molly and Olivia both are agriculture teachers, we were talking a lot about how these things can transfer into an agricultural classroom, things like aquaponics, hydroponics, talking about jobs and business, and how to actually, one of the more interesting things that we talked about, I thought, was being able to do micro, like, hydroponics kits where you could do a tabletop one and raise like pea shoots, so that you would have greens in, and Olivia was talking about how that she was doing that with kids, thinking about how people in New York City could get fresh produce during like COVID, for example. And then we kind of just reflected on the series and how things went. There was a general consensus that things went well and people got things out of it, which I thought was great, considering that I don't think, at least because my school doesn't offer an agriculture or business program, I hadn't thought that, really to point this workshop series toward that. So it was cool that people who were from other areas of content, not just science teachers, were able to pull really meaningful things from the series. And I thought had a really great idea about like a Great Lakes 101, like broad offering of some sort to just kind of level set and figure out, okay, here are the issues, the current issues surrounding the Great Lakes from, the perspective of agriculture or business or, even geology, and not just biology, but some of the other things that are influencing and affecting the Great Lakes, even politically, would be a great way to kind of launch something like this. Because I think one of the things that I noticed as I was in breakout rooms was that we didn't have just science teachers this time. And I think when we do the shipboard science stuff, we have a lot more people who are, chemistry, physics, and bio, or they're elementary school teachers who have a bent that way. This time it seemed to be that there were a lot more like art and English and ag teachers than I've seen in Sea Grant programming. How'd I do? Was that okay? All right.
I would like to just add one thing. Sorry, I didn't mean to interrupt you, Marty. Just that Olivia mentioned too, wanting some assistance on being able to provide who was it? Celine said about having those packs ready to go for the teachers to give to their families remotely. So maybe it's something Sea Grant can talk with you, Celine, on how you did it, or what kind of resources, materials you gave. That would be really helpful, and then we could share that with the rest of the folks that are participating in the workshop. So sorry, Marty, didn't mean to.
No, that's just fine. So I guess that leaves our last group here. And Lori, I'm going to ask you again, since you're the one that kicked that off. Do you have those notes in front of you? Are there things that you want to share from that, or should I just go ahead and take the?
I think you can just go ahead. I'll jump in at the back end if there was anything that I remember.
Okay, that sounds good. So we just started sort of working through some of the questions. We did talk briefly about the action plan and how there's already some ideas in place for collaborating over the course of the year. Lori and Brian are interested in collaborating on some work, and so we'll be curious to see how that comes to fruition. It sounds like it was a thing that had begun in the past, but then because of our life changes, it's something that will be taken up a little bit later. And of course, Lori could talk about that better than I can, so I will not say any more about that. We also talked a little bit about some of the technical aspects that were introduced during this workshop series. Things from, just learning how to do things like take images and upload them, and things like that are some skills that maybe educators at this point are now, if they don't have them already, are really interested in learning how to do because of this new online world piece. We also had a really interesting point that was made about just sort of looking at our workshop structure overall and seeing what might be able to be used and pulled into teaching for the 2020-2021 school year. And so some of the pieces that were most identified as valuable for maybe bringing back to the classroom from a workshop structure standpoint are the macroinvertebrate pieces, where students are sent out to do something and then can be brought back in, and having students figuring out a way to use a technology to do identification and things sort of like we did today. Some of the topics that were most interesting, in addition to the macroinvertebrates, was there was a lot of interest expressed in the winter ecology piece, just because school, of course, spans winter time. And so the ideas that of ways to get students out and things to engage in over the course of the winter was valued. by the folks that I was working with, and I was working with Brian and Dave and Laura for a little bit, and MJ and John. So everyone there kind of felt that those were good pieces that occurred because of the workshop. In general, the workshop structure was really favored. Joyful hours were a great thing. One of my notes just says, keep them, and maybe they're a little too short actually, because the time just flies. There's always that balance between getting everything done that we want to do and still respecting people's time. So that's always a challenge that we have. And gosh, there's a lot more, but we only have a couple of minutes left. So let's see if I can pick out some other nuggets here. Oh, one of the pieces of feedback that we talked about was that having the assignments given ahead of time and having that Google folder and the structure that happened before the workshop actually happened was really valuable. So those are pieces too for the future. John Weemholt just dropped in a list in the chat, and also in things like, I sort of got a feeling of a theme developing about just water in general, all the way from like the local level in the back stormwater ponds of your school, all the way out to the oceans. So we just need to cover all the water is kind of what I heard. And I think that's what I'm going to stop, because we only have a couple minutes left, and I want to make sure that if there's anything else that someone wants to say, that they have an opportunity to do that. So any other last thoughts or comments that anyone has?
You all put in a huge amount of work. We're all teachers. We know how much prep work is required to pull off something like this. And I would just like to say thank you for all your hard work and all the hours that you put into it. It was very, very valuable.
I just want to say that I've had a heck of a lot of fun being part of this. So I think all of us feel that way. It's been really good for us and wonderful to see you guys sticking it out week after week in a really tough year. So thank you.
Yeah, we knew we were onto something when people kept coming back. So, and we had a really great planning team. And so that I think made things so much better. We had a good rhythm sort of set up. And I said this in my small group, and maybe this is where I'll close it for now, is that one of the limitations for the virtual workshop series is potential limitations is we really enjoy and value the small group work. And because we really value and enjoy the small group work, but we need people who can facilitate in those areas, that's sort of the pinch. And that's where we look to teachers who now have gone through the virtual workshop series to consider coming back and assisting with some of those pieces. Because if we want to continue doing this sort of format, that's where we really need, I think, the assistance is in the group facilitation for the small, meaningful conversations that we have. So put that in your cap as you're thinking through everything. And I just want to say thanks to everyone for coming, and I want to say just the best of luck for this coming school year. We will be thinking about you a lot, and keep in touch. We will see you again. Now that you're here, we'll see you again. So, and I think that's it. Here ends the workshop.
Thank you. Bye. Thanks for everything, all your participation.